Bismillahirrahmanirrahim. Today we will discuss regulation of the blood pressure, how the blood pressure is regulated in our body, what are the mechanisms for it. So the learning objective for this lecture are you have to identify what are the factors affecting the mean arterial pressure, the concept of different regulatory mechanisms that play a major role in controlling of the blood pressure. Some are acting very fast, some are acting very slow. So based on that, they are classified as short-term and long-term mechanisms. In that, we will discuss in detail about the baroreceptor mechanism, which are one of the short-term mechanism in detail. And we will also discuss one long-term mechanism, which is known as the renin-angiotensin-aldosterone mechanism, which is a type of long-term mechanism. Then we will see what is the importance of short-term and long-term mechanisms of regulation of the blood pressure in our body. Then the describing the normal values of systolic and diastolic blood pressure, and based on that classifying it to be as hypertension or hypotension, and what are the guidelines. So first of all, define blood pressure. What is blood pressure? Blood pressure is nothing but the pressure which is exerted on the vessel wall. Suppose this is a blood vessel and the blood is flowing in it. So the pressure which is exerted on its wall is known as the blood pressure. So it is a lateral pressure exerted by the column of the blood on the vessel walls is called as the blood pressure. It depends upon two factors. It depends upon two factors. How much blood is present here, that is the volume of the blood, and how stretchable or compliance of these vessel wall. How stretchable these vessel wall. If these vessel wall becomes very stiff, then the pressure will increase. If these vessel wall are soft and stretchable, the blood pressure will remain or regulated normally. Then we have classified the blood pressure into two types. The pressure during the ventricular systole, or it is called as the maximum pressure or peak pressure during the ventricular systole, is called as the systolic blood pressure, which is recorded as for the purpose of understanding. We call it as in a young adult person ranging between 100 to 140 millimeter of mercury. We take average as 120 millimeter of mercury. Whereas diastolic pressure is the least or minimum pressure during the diastole of the ventricles. Ventricular diastole, what is the minimum pressure in the circulatory system or in the vessel, is called as the diastolic blood pressure. It also ranges between 60 to 90 millimeter of mercury. 60 to 90 millimeter of mercury. And for the purpose of description or understanding, we take average as 80 millimeter of mercury. And if you take systolic and diastolic, then the difference between the systolic, let's say systolic is 120 and the diastolic is 80. This difference is known as the pulse pressure, which is around, in this case, it is about 40 millimeter of mercury. So difference between systolic and diastolic is known as pulse pressure, which is you can feel the pulse, where the pressure when during the systole increases and it touches to your fingertips, and it goes off during relaxation or during the diastole. That's why this difference is called as a pulse pressure. What is mean arterial pressure? Mean arterial pressure is nothing but the average pressure. It is the whole, not during systole, not during diastole, but on an average in the whole of the circulatory system. How much is the pressure present or found in the circulatory system is called as the mean arterial pressure. So based on this mean arterial pressure, we will describe what are the mechanisms which are controlling the blood pressure. So the mean arterial pressure is calculated by knowing the systolic and diastolic. So the formula for mean arterial pressure is diastolic pressure, let's say 80 millimeter of mercury, and 1/3 of the pulse. 1/3 of the pulse means you have to first find out the pulse pressure, which is nothing but the difference between systolic and diastolic. So with a value of 120 over 80, if suppose the person is having systolic blood pressure 120 and diastolic is 80, his mean arterial pressure will be diastolic, that is 80 plus one third of 40. One third of 40. If you say, let's say for the purpose of calculation, let's say it's not 40, it is one less, that is 39. So that we can divide it into three equal half, three parts. So 80 plus one third of 39 is 13. So 80 plus 13 is 93 millimeter of mercury. So this is called as the mean arterial pressure. This is for calculation, but in general it is an average pressure present throughout the circulatory system irrespective of systole or diastolic condition of the ventricle. In our body, the blood pressure is mainly because of, or it depends upon, what are the primary determinants of the blood pressure are cardiac output, how much blood pumped out by the ventricle in 1 minute, this amount, plus the, the resistance which is felt for the flow of the blood, total peripheral resistance. These are the two main determinants which is describing or deciding what should be the mean arterial pressure. So we can say that mean arterial pressure depends upon cardiac output as well as total peripheral resistance, TPR. If you remember, half of this picture is already discussed with you. What was that? So as I told you now, that mean arterial pressure, or it is written as MAP, depends upon what? Two factors. One is cardiac output, second one is total peripheral resistance, the resistance offered to the flow of blood in the periphery, in any part of the body. On an average, what is the resistance felt to the flow of the blood is called as total peripheral resistance. Now this part, the left part, we already discussed in the previous lectures of cardiac output. Now cardiac output depends upon what? It depends upon the heart rate, how many times the heart beats, and it also depends upon how much blood is ejected out during one beat, that is called as the stroke volume. So cardiac output depends upon stroke volume as well as on the heart rate. Now if you remember, the heart rate depends upon, or it is regulated by sympathetic and parasympathetic stimulation. So sympathetic as well as parasympathetic will regulate or change the heart rate. Whereas stroke volume, the amount of blood ejected out per beat, it depends upon how much blood is collected, which is nothing but the venous return. If venous return becomes more, the blood collected in the ventricle will be more and the stroke volume will be more. And if you remember, venous return, the blood which is returning to the heart, depends upon many factors. What are these? If the blood volume is more blood will return to the ventricle. If the person is having strong respiratory activity, deep and forceful respiration is going on, there will be more venous return because it will create negative pressure in the chest cavity, which will pull the blood towards the heart. Also, if the person is doing muscular activity, exercise, and the peripheral blood will be shifted towards the heart, that is the venous return will increase. And also during the phases of contraction and relaxation, there is a negative pressure developed within the heart chamber, which is called as the cardiac suction effect. This will also pull the blood towards the heart, that is venous return will increase. So all these factors are affecting the stroke volume. Ultimately it is affecting the cardiac output, and that's why it will affect the mean arterial pressure. So we can say the primary determinants of the primary determinants of mean arterial pressure are cardiac output and total peripheral resistance. But it is indirectly affected by heart rate, also by the stroke volume, and it is indirectly affected by sympathetic parasympathetic stimulation, and indirectly affected by the venous return, respiratory activity, skeletal muscle activity, cardiac suction effect. All these factors are affecting stroke volume and heart rate, ultimately affecting the cardiac output, and they are affecting the mean arterial pressure. So whenever there is a change in the mean arterial pressure, you have to think about all these factors. Is there any increase in the cardiac output, or is there any increase in the heart rate, or is there any increase in the stroke volume, or is there it is sympathetic stimulated or parasympathetic stimulation? So indirectly you have to think about many factors while determining the value of blood pressure as normal or abnormal. Similarly, total peripheral resistance is affected by what? By blood viscosity. If the blood is thick, resistance will be more, and it depends also upon the size of the vessel, or you can say the diameter or radius of the vessel. So if the radius is less, the resistance will be more. If it is dilated, the resistance will be decreased, and we have seen the relation is not direct, it is not 1:1. It will be the fourth power of the radius. It is fourth power of the radius. If it is r raised to 4. Arteriolar radius is frequently affected by the metabolic content in or around the vessel. Also, it is affected by sympathetic stimulation, either sympathetic stimulation or the effect of epinephrine on the blood vessels. Blood viscosity depends upon the number of RBCs. If there is a polycythemia, blood viscosity will be more, total peripheral resistance will be more, mean arterial pressure will be more. The other factors which is affecting the vessel radius is vasopressin, release of hormones in the periphery like vasopressin and angiotensin. They cause this usually vasoconstriction and leading to increased peripheral resistance. It is also, these two factors are also affecting on the other side, that is vasopressin, renin, angiotensin system is there, which is responsible or acting through the kidney to absorb more salt as well, and that's why increasing the water along with it, and that's why it will increase the blood volume. So all these factors are affecting the mean arterial pressure. This is another elaborate view of showing that how the total peripheral resistance will be affected. It is nothing but it is related to the arteriolar radius. It is affected by the local metabolic control like presence or absence of or increase or decrease of CO2, O2, hydrogen ion concentration, potassium, adenosine, ATPs. All these are factors which is causing uh, the change in the arteriolar diameter. Similarly, extrinsic control which is coming from outside are nothing but the hormone like vasopressin, angiotensin, and epinephrine. It is affecting the arteriolar radius. Viscosity is affected by the number of RBCs. Sympathetic activity also affect the arteriolar radius, and so these are the two main factors: sympathetic plus the metabolic. You can say autonomic and metabolic. So basically, regulation of blood pressure means regulation of cardiac output as well as regulation of the total peripheral resistance. So the body is trying to keep the balance between the cardiac output and the peripheral resistance, and that's why it is uh, making the mean arterial pressure remain within the normal range. If these two factors are regulated, that means mean arterial pressure will be regulated. So we have to see what are the mechanisms which are regulating cardiac output and what are the mechanism regulating the mean arterial pressure. Mean arterial pressure or the blood pressure is constantly monitored by some sensors in our body. There are pressure sensors in our body. We call them as baroreceptor. Pressure means baro. Receptors are nothing but the sensors. So baroreceptors are nothing but the pressure receptor present at a strategic point in our body which are monitoring the pressure. Okay, so short-term regulation of the blood pressure is through the baroreceptor. Why it is called as short-term? Because it can regulate the blood pressure within seconds. Within seconds, suppose the blood pressure changes from lower to normal to high, then within seconds it will be brought back to the normal. That's why it is called as the short-term mechanism. It will take only few seconds to revert the change, any change. Suppose it is lower, it will make it normal. If it is higher, then it will also become normal within seconds. That's why it is called as the short-term mechanism. And it is mediated by, how it is mediated by the autonomic nervous system. So there is a great role of autonomic nervous system in regulating the blood pressure through short-term mechanism. While there are other mechanisms which starts little late when the blood pressure changes. Let's say blood pressure is 120 and it becomes 160. Then the mechanisms will start to bring it back to 120, but it will take time, maybe few hours, minutes, or 2 days, maybe. So that's why it is called as the long-term mechanism, not like the short-term, which takes only few seconds, which take only few seconds to bring or regulate the blood pressure. So what long-term mechanism will do? Long-term mechanism involves adjusting the total blood volume. It is mainly related to the blood volume and to the salt and water balance in our body. Salt and water balance in the body. So it is done through kidneys. It is because salt and water balance is done through kidneys. So kidneys play a role in regulating the blood pressure. Okay. We will discuss one short-term mechanism, which is baroreceptor, in detail to understand what is the meaning of short-term mechanism and the role of kidney as a long-term mechanism. Rest other we will just see the few slides. Okay. So what are baroreceptors? Baroreceptors are nothing but the stretch receptor. What do we mean by stretch receptor? That means these are the receptors whenever they get elongated or brought back to the normal, the initial position, just like a elastic tissue. Initially it is in this length is this much. It may be getting stretched, becoming long, or it may become very short. So stretch receptors, based on the pressure, it can be elongated or brought back to the normal. They are present in the blood vessel wall. They are present in blood vessel wall, specifically in carotid sinus and aortic arch. Carotid arteries which are supplying blood to the brain, to the head region. Here the arteries which are going there, we call it as carotid arteries, carotids. And also they are present at the aortic arch, arch of the aorta. So in this region, these stretch receptors are located. In this region, it is stretch receptors are located, and they are monitoring the change in the pressure. That's why they are called as the baroreceptors. Here is the picture showing you that at the bifurcation of the common carotid artery, in the internal carotid artery, area becomes slightly elongated or become distended. We call it as carotid sinus. Here these stretch receptors are located. We call it as baroreceptors. Also in the wall of the arch of the aorta, stretch receptors are located. We call them as baroreceptors. So they will sense, they will check whether the pressure is increasing or decreasing from the normal. Any change, either increase or decrease, will be sensed by these receptors here and here. And whom they will inform once they detect any change? What will happen? They will take the signal or the information to the higher center, the center which is regulating the blood pressure. So baroreceptors are detecting any change, either increase or decrease in the pressure, and they are working through the autonomic nervous system and informing to the cardiac center. Where the cardiac center located, we will see it here. And reflexly, it will bring back the pressure to the normal. So there is, as the name suggests, it is a reflex mechanism. Reflex. That means something is happening in that event. It is bringing back the change to the normal. This is called as a reflex. Once, let's say the example, if something is coming in front of you, in front of your eyes will blink automatically without any thinking. This is called as a reflex mechanism. Or if you put your hand on a hot plate, suddenly you will lift your hand, because it is a reflex mechanism that without knowing, or conscious information, your hand will be pulled back from the hot surface. This is called as a reflex mechanism. So for any reflex to occur, there are certain components are required. What are these components? There should be a sensor, we call it as a receptor. There should be a nerve which is taking the information to the spinal cord or any center, that is the center. And then there is another nerve, or which will bring the information back to the effector organ. It may be a muscle, it may be a gland. So these are the components. One is the receptor, which is nothing but a sensor. Then the carrying by a nerve, which is called as an afferent pathway, because it is going towards the heart, towards the center. That's why called as afferent. And there is a third one, which is where it will take the center, and then the information is coming out, exit, from the nervous system, or from any from the center to the periphery. It is called as efferent. And then the effector organ. So all these components are in a form of arc. Start from point 1, this is 2, this is the center, 3, this is the efferent artery, 4, not artery, it is efferent nerve or any pathway, and the effector organ, 5. So all these are made in a fashion of an arc. Arc means horse. In Arabic, it is called as horse. So it is a reflex arc. So receptors are located in carotid sinus. We have seen an aortic arch, and they are carried through the nerves. The information is carried in the form of action potentials. How the information goes to the nervous system? In the form of action potentials to any change in the pressure. Here it is. It is explained here. The afferent pathway, which is carrying the information from carotid sinus, is the ninth cranial nerve. Is the ninth cranial nerve, also known as the glossopharyngeal nerve. And from the arch of aorta, the 10th cranial nerve, which is known as the vagus nerve, which will carry the information to where? To the cardiac center, which is located in medulla oblongata, brain stem. It is the part of the brain stem. If you remember, this is the cross section. Here is the brain, and there is a pons. This one is the pons, and this is a medulla oblongata. So in this area, there is a center, we call it as cardiac center. Here the information is coming in the form of nerve signals, okay, which are in the form of action potentials. Then what the center will do? Suppose the center find that blood pressure is raised to, let's say, 160. Systolic blood pressure is increased 160. This center will send the information in the form of a output signal in the form of autonomic signals or sympathetic signal to the effector organ, to the heart as well as the blood vessels, to either slow down or decrease the cardiac output. I will show you the example here. This is another way to explain you that there is a loop, or there is an arc is forming between the arch of aorta and the carotid sinus, and then information going to the medulla oblongata, and from there the response is coming back to the effector organ. So this is how the whole uh, reflex arc is functioning. baroreceptor reflex. To explain how this system works, we have to think about two conditions. In one condition where the blood pressure is increased. When the blood pressure becomes elevated, what is normal? Normal is up to 140 millimeter of mercury, okay? Systolic, and 90 millimeter of mercury diastolic, it is the maximum level, okay? If suppose it is more than that, let's say it becomes 150, or diastolic becomes, let's say, 95 millimeter of mercury, what will happen? This is detected by the carotid sinus. The stretch receptors becomes more stretched, and they will start to increase the action potential generation. They will increase more action potentials. This action potential will travel through the 9th and 10th cranial nerve to the cardiovascular center in the medulla oblongata. Medulla oblongata receives the information that the pressure is elevated. What it will do? It will cause the response opposite to the initial one, that it will decrease the sympathetic activity, that is decrease the vasoconstrictor activity of the blood vessels, and it may increase the parasympathetic activity. That's why the heart rate becomes slow, the stroke volume will become low, and there will be increase in the arteriolar diameter or vasodilatation will occur. In total, there will be decrease in the cardiac output, and there is decrease in the total peripheral resistance. And because of that decreased cardiac output, decreased total peripheral resistance, blood pressure will come back to the normal within 120 to 140 systolic. It will come back to the normal, or between 60 to 90 diastolic. So the initial, it is increased here, detected by the baroreceptor, have done some actions, and these actions will resulted into the pressure come back to the normal. So this is in elevation condition. What will happen if the blood pressure is decreased? Let's say it is less than 100 systolic, or less than 60 diastolic. If it is like this, then the carotid sinus and aortic arch will detect there is less pressure. Because of this less pressure, the action potentials which were produced in these nerves will be reduced. They will be less than the normal, and this information is reaching to the cardiac center, cardiovascular center, in the form of less action potentials. Because of this, the cardiac center, what it will do? It will increase the sympathetic activity and decreases the parasympathetic signals. This will lead to increase in the heart rate, increase in the cardiac output, stroke volume, and it will cause vasoconstriction effect. This will cause peripheral vasoconstriction, and that's why increased cardiac output, increased total peripheral resistance, so blood pressure will come back to the normal, come back to the normal level. So in the both conditions, increased pressure or decreased pressure, the baroreceptors are functioning as a normal, okay, to normalize the blood pressure, and this is happening within few seconds. That's why it is called as the short term mechanism. Short term, okay. To understand what is short term, within how many seconds. Suppose the person is in the supine position, sleeping. Maybe he or she is not feeling well, sick, and sleeping for a long time, maybe more than 12, 13 hours, and then suddenly woke up. Not only woke up, but stand up directly. Is not sitting down. From the lying down position, directly the person becomes erect. Because of this, the blood supply to the head decreases because of the gravity effect. This will cause decrease in the cardiac output, and this will cause decrease in the arterial pressure. This is detected by the baroreceptor. Suddenly the baroreceptor will will receive that there is a decreased pressure, and it will inform to the vasomotor center, which will cause increased sympathetic activity. And this increased sympathetic activity will cause the pressure which was decreased now come back to the normal. come back to the normal. This is how the baroreceptor function. So baroreceptors are functioning within seconds. Suddenly you woke up and stand up without feeling dizziness, the pressure becomes normal. This is called as the baroreceptor mechanism. Maintain, it is the function is to maintain relatively constant pressure despite you change your posture. Sometimes you are sitting, sometimes you are lying down, sometimes you are standing, jumping, still the pressure will remain constant. This was proved by experiment done on the dogs. What they do in dogs, they cut these nerves. Which nerve? Ninth and 10th cranial nerve. Before cutting the nerve, they observed that the blood pressure ranges between a narrow range. It is ranging between maybe 90 till 110. Once they cut these nerves, the one which is taking the signals to the cardiac center, they found that blood pressure is start fluctuating between very low to very high, very low to very high, because they are not sensing it, they are not detecting it, and that's why the pressure will be variable throughout the day, minute to minute it is changing. Same thing is shown here. This red color graph is about after cutting the nerve, that is denervated. When the nerves were intact, the blood pressure was in a narrow range, in a very small range. But once the nerve is cut, the fluctuation is seen in the blood pressure. One more thing is that if the blood pressure remains very high for long time or very low for long time, these receptors get adapted. They will not respond. They will respond only to the variation. Whenever the blood pressure is changing, these receptors are quickly performing their action. But if the blood pressure is high for maybe 1 week or low for maybe another week, then this receptor will set on that lower or higher pressure. That's what they get. Baroreceptors get adapted to a higher or lower pressure. Okay, apart from this short-term mechanism, this is one example. Another example of short-term mechanisms are example is peripheral chemoreceptor, central chemoreceptor, and cardiopulmonary baroreceptor. These are also the regulatory mechanisms which are functioning as short-term mechanism, but they are not described in detail. We will just go quickly on each of it. What is first one is cardiopulmonary low pressure receptor. These receptors are, as the name suggests, cardiopulmonary. They are present in the heart as well as in the vasculature of the lung. They are present here. These are the stress receptor again. They will sense the change in the volume. They are sensing the change in the blood volume or fullness, increase or decrease in the volume of the blood in the circulatory system. They are located in atria as well as in the veins and pulmonary artery. So if there is an increase in the blood volume, suppose instead of 5 liters, suppose the blood is increased to 6 liters, then these receptors bring the following changes. The if the volume increases, what will happen? The more blood will be received in the atrium, and the atria get stretched, and there will be secretion of one hormone from the atria which is known as ANP. The full form of ANP is atrial natriuretic peptide. Natriuretic peptide. Atrial natriuretic peptide or ANP is released. This is also increased volume is also causes decreased secretion of ADH, antidiuretic hormone. Antidiuretic hormone. These are the hormones which will which will be affected by the volume, and it will also cause renal vasodilatation, and it will cause increased heart rate. So due to, if there is an increase in the blood volume, these receptors bring about following changes. Atrial natriuretic peptide will be released. What it will do? It is coming from the atrial muscles or from the atrium. It is its action is natriuresis. Natriuresis means loss of sodium in urine. So increased secretion of atrial natriuretic peptide will cause loss of sodium from the kidneys in the urine. More and more sodium will be lost. Whenever sodium is lost, water will also be lost. So that's why the increased pressure or increased volume will be compensated by losing the more and more sodium and water, and the blood pressure will become to the normal. Also, it causes release of ADH, anti-diuretic hormone. Diuresis means formation of more urine, increased volume of urine. Diuresis is increase urine volume. This is called as, this hormone is causing antidiuresis. Antidiuresis. And its concentration decreases. Antidiuresis means the one which is conserving water. It will absorb more water. So if its concentration decreases, there will be decreased conservation of water and water will be lost in the urine. Again, in the urine, there will be loss of water, more formation of urine. It is also causing renal vasodilatation, so more urine formation, and it causes increase in the heart rate. All this is happening in a very short period of time. That's why it's called as a short-term mechanism. Chemoreceptors, as the name suggests, these receptors or these sensors, they will detect chemical change in the blood. Chemoreceptors, these are the receptors which will sense or detect chemical change in the blood. What chemical change? This is the chemical change. Change in the oxygen concentration, change in the CO2 concentration, or change in the hydrogen concentration or the pH. pH is nothing but the hydrogen concentration. Any decrease in CO2, any decrease in oxygen or increase in CO2 or decrease in the pH, or let's say increased hydrogen concentration, will be detected by chemoreceptors. And these chemoreceptors are also located in the arch of aorta as well as in the carotid arteries, and they are placed at carotid bodies and aortic bodies in the form. And they will inform to whom? To the vasomotor center. Vasomotor center, another name for cardiac center. Vasomotor center. This will lead to increased sympathetic activity and blood pressure will be raised. So if decrease O2, increase CO2 is happening, the blood pressure will increase. This is called as chemoreceptors, and these are central. But the important thing in this is that these receptors or chemoreceptors are not stimulated until the mean arterial pressure falls below 80 millimeter of mercury. Unless the 80 millimeter is reached, they are not functioning. If it is more than 80, they will not function. If it is less than 80, they will start acting or functioning. Similar to that, central chemoreceptor CNS ischemic response is another example of short-term mechanism, and it is related to the oxygen concentration to supplying to the brain. Okay, so whenever cerebral blood flow decreases, it will cause CO2 to be built up there, accumulation of CO2 in the central nervous system. This usually happen in the form of decreased blood flow, which is called as CNS ischemic response. So cerebral ischemia occurs, cerebrum gets less oxygen, decrease oxygen to the cerebrum, or increase accumulation of CO2 there. So vasomotor center will be activated, and there will be increased sympathetic activity, and the arterial pressure will increase, and the oxygen supply will also be coming back to the normal. Again, the CNS ischemic response will not be active unless the mean arterial pressure falls below 60 millimeter of mercury. So you have to remember that baroreceptors are functioning at normal pressure, like uh, mean arterial pressure of 90, whereas chemoreceptor, they are acting at lower pressure, like the 60 millimeter of mercury. Prolonged CS ischemia, if it remains, then it will lead to depression of the vasomotor center. If it is for long time, if there is CS ischemia, then the vasomotor center will also be depressed and not function. Coming to the renin angiotensin aldosterone system, or is an example of the long-term mechanism of regulation of the blood pressure. We have seen the baroreceptor, chemoreceptor, and CNS ischemic response, and the atrial cardiopulmonary low pressure receptor. These are all examples of short-term mechanism, which is quickly acting and quickly getting adapted. But here is the example, another example is the long-term mechanism, when the blood pressure is not regulated, or it is elevated or depressed for long time, and the body will start to function, or body will start to compensate it, but it will be responding in a late fashion, maybe starting after 20 minutes and becoming active, starting in few minutes and then becoming active, more fully active in 20 minutes to few hours. This is called as long-term mechanism. Example is renin angiotensin system. What is renin angiotensin system? Renin is an enzyme. Renin is an enzyme released by the, from where it is coming? It is coming from kidney. In response to what? In response to decreased blood pressure. If the blood pressure becoming low, and it is detected by the kidneys in the form of release of renin, in the form of release of renin. From where the renin is coming? Renin is coming from a special type of cell in the kidney, which is known as the juxtaglomerular cells. Once it is formed there, it will enter into the blood. It will come into the circulation, and it will act on another substrate which is called as angiotensinogen. So from the kidney, renin is coming, which is coming in circulation, and in circulation it will find that there is angiotensinogen. Renin will act on angiotensinogen to form angiotensin 1. Angiotensin 1. Now you may be asking from where the angiotensinogen is coming. Angiotensinogen is coming from, it is a peptide substrate, it is coming from liver. It is coming from liver. So here it is, how the renin angiotensin system function: decreased blood pressure, arterial pressure, leading to release of renin from the kidney. So kidney will release renin. This renin will act on a substrate called as angiotensinogen, which is coming from, formed by the liver to form angiotensin 1. Once the angiotensin 1 is formed, it will go to where? To the lungs, because again it is not active, and it requires an enzyme which is present only in the lungs, which is known as angiotensin converting enzyme. Angiotensin converting enzyme, ACE, which is present in the pulmonary blood vessels to form the angiotensin 2. This angiotensin 2 is very active. It is causing renal retention of salt and water so that the blood pressure will become normal. It will also cause vasoconstriction. So again, the total peripheral resistance will increase and blood pressure will become normal. Or it can be deleted, or you can say destroyed, by the angiotensinase enzyme so that it will not function continuously. Ultimate result is the blood pressure will come back to the normal. So this is a long-term mechanism use of or with the help of kidneys. Same thing is shown here, that liver synthesizes angiotensinogen. Kidney, in presence of low pressure or in presence of low oxygen there, it will release more renin. This renin will convert angiotensinogen to angiotensin 1. Then angiotensin 1 will go to the lungs, because there is an enzyme called as ACE, angiotensin converting enzyme, which will convert it into angiotensin 2. This angiotensin 2 will act on adrenal cortex to release aldosterone. Aldosterone is the hormone which will regulate the sodium concentration. So that's why the role of kidney is there, which is causing retention of or absorption of sodium as well as water. This will lead to increased blood pressure. This angiotensin 2 also act on the nervous system to increase the thirst mechanism. So person will drink more water and the volume will become more. This is also acting on another enzyme which is called as vasopressin, which will cause, or another name for vasopressin is ADH, antidiuretic hormone, which will increase the absorption of water. So ultimately it is increasing the blood pressure. Okay. So this is a long-term mechanism of the regulation of blood pressure. Now up till now we have seen blood pressure can be decreased or increased. If it is increased more than the normal range, that is, we say maximum range is about between 100 to 140 systolic and 60 to 90 diastolic. If it is more than that, we call it as hypertension. If the blood pressure is below the normal range, less than 100 systolic or less than 60 diastolic, we call it as hypotension. So hypertension is increase in the blood pressure, hypotension is the decrease in the blood pressure. Nowadays, the doctors or different organization, they said that anything below 120 systolic is normal blood pressure, anything below 80 millimeter of mercury is the normal diastolic blood pressure. Anything between 120 to 139 is pre-hypertension. That means it may land into increased blood pressure. Or anything between 80 to 89 diastolic is pre-hypertension. Whereas 140 to 159 is stage 1 hypertension, or 90 to 99 diastolic is stage 1 hypertension. More than or equal to 160 systolic or more than or equal to 100 diastolic is stage 2 hypertension. But whatever may be the stage, if anything above the normal is called as hypertension and below the normal it is called as hypotension. Another way of classifying hypertension is hypertension can be of two types. It can be primary hypertension or it can be secondary hypertension. Before we go to the first one, I will say this secondary. Secondary means the hypertension or increased blood pressure due to some other disease, other cause, other problem or other disease, like nervous system disease or CNS disease or endocrine disease or renal disease. Because of this disease, if the blood pressure is high, we call it as secondary hypertension. And if there is no other problem except increased pressure, it is called as primary hypertension. So primary hypertension means 90% of the cases of hypertension are of primary type, where no other cause is found except increase in blood pressure. They say there are so many theories are there. They say hypothesize that it could be due to defect in salt management, that is kidneys are not functioning properly, can lead to increased blood pressure, or the person may be taking high salt intake in the food, or it may be taking low potassium or low calcium in the food or in the diet. Then there may be plasma membrane abnormality. which is mainly related to abnormality of sodium potassium pump, can lead to increased blood pressure, or can be problem related to angiotensinogen or any genetic disorder. There could be digitalis like substances in the body, or abnormality of nitric oxide, endothelin, which are causing vasoconstriction and vasodilatation effect. They may be affecting the blood pressure, or excessive vasopressin. So primary causes are, there are so many theories are there, not proven. We have to think that it could be anything of it. That's why it is called as primary hypertension. We don't blame any other organ. Whereas secondary hypertension is because of it is around 10% of the cases suffer from secondary hypertension, and it could be due to some other problems like renal disease, endocrine disease, or nervous system disease leading to increased blood pressure. This is called as secondary hypertension. If suppose the blood pressure is remain high for long time, what are the damages can occur in our body? It could be damaging to our heart, leading to congestive cardiac failure. It can lead to stroke, that is the nervous system. There may be either decreased oxygen supply, or there could be uh, for blood coming out, hematoma is forming around the brain. So it could be leading to abnormality of the CNS, stroke, or it can lead to heart attack. It can lead to spontaneous hemorrhage anywhere in the body. It can lead to renal failure. It can lead to retinal damage, that is blindness. It may cause damage to the retina, which is the light sensitive layer of the eyeball. It can be damaged because of the increased pressure for and not regulated blood pressure. So blood pressure has to be maintained in a normal range by either by mechanisms in the body, short term or long term, or by using the antihypertensive drugs. Otherwise, the person may suffer from these abnormalities. With this, we come to an end of our lecture of how the body regulates the blood pressure regulation mechanism, short term and long term. Examples of short term, we have discussed detail, the baroreceptor. Then we described other short term mechanisms like chemoreceptor, low pressure receptor, and we described the long term mechanism as how the kidneys help in regulating the blood pressure, which is for long term. It takes long time to activate, and uh, it will be activated uh, after maybe 20 minutes to few hours. So these are all about regulation of blood pressure. Thank you very much.